So much fun. So much fun. I'm having so much fun spending money. Love it.
Crying. Okay. Should we wait till the O5 to see if people will come?
I think we'll give folks another maybe minute and then they can just go from the recording otherwise.
Okay.
Especially because we're working with the data today, so I want folks to actually have enough time to work through that with you.
By the way, Adina, were you able to look at the? Technically, this would have been like less than a working day of time, but were you able to look at the recording to see if you're able to open the documents within Python in your Jupyter Notebook setup?
Yeah,
Oh, nice. No
I
issues?
did get
Or
some
like,
help. No,
okay,
no,
good.
yeah, I got me and Alicia ran through it. Yeah.
Okay, good. Yeah. That is good, at least that you guys are able to help each other. Because, like I said, like it's the same issues that you typically run into, and a lot of the language is very confusing. Like I happen to know this because this has been my job for like the last 10 years, but especially when I started, like I had no idea what I was doing or how to install these software. So it would make sense for you guys to struggle solo or spend 5 hours trying to fix something that we could fix in an hour.
Also, sorry I didn't send a paper into the chat. I did find like I was like browsing through a couple, but one that I kind of landed on was called Efficacy of Probiotic Supplementation in Reducing Primary Dysmenorrhea, a double-blinded randomized controlled trial. I was looking for things that were like had to do with like women's health. Um, so yeah, I just found that paper, but I actually need to like I do want to still read it. I know it's a little past time for that, but
That's okay.
yeah.
In general, it's just good to have the practice of it. And then also, like, the reason for sending them in the chat is so that we all know what we're each interested in and so that I know what's interesting to you so I can try and integrate it more into our upcoming weeks. My PhD work was in how the microbiome changes during pregnancy. And so I have some background in vaginal microbiome and in women's health, but I've been out of that field for like 2 years now. But it is an interesting field and it's like more controversial than it should be. But it's a potential promising field with therapeutics and stuff because, for example, like a lot of women suffer from BV or bacterial vaginosis. And this is just a very vague clinical syndrome in which they'll give you a checklist. And if you check a certain number of things off of this checklist, they say you have BV. Or they will basically like do a swab to look at the bacteria that are growing inside of the vaginal environment. And if you get a certain number of bacteria that look a certain way, then they'll give you the diagnosis. But sometimes healthy women also have those bacteria, but they don't have those symptoms. So it's not clear like what's actually causing the disease. Like maybe the microbes are there, but they're not a marker of disease. And so it's really interesting because it's just like an area of study where we're very actively changing our understanding of what's happening in the body to try to understand the disease. My cat's biting my electrical wires.
It's such an interesting area of study. What I find frustrating, I guess, is the lack of like a sparsity of data when it comes to women's health in general, which really does make it more difficult to build kind of like more robust studies. So the kind of exploration that you might be easily able to do in things like cancer, just because of the amount of data present, is oftentimes not something that we can do because the data sets are so incomplete. And often that's due to funding and what gets funded. So I know that at Yale, there's also, in my opinion, kind of like a lack of laboratories that specifically focus on things like, the vaginal microbiome or even pregnancy-related kind of pathologies are definitely underrepresented.
I mean, in general, like being pregnant in the US is really scary because like everything else the same, the US has really bad maternal health outcomes and pregnancy complications, especially like I mean, what the most high profile cases I think in recent years were like Beyonce and Serena Williams. I think Serena Williams almost bled out and died while she was giving birth, and she was like in a hospital,
Yeah,
in a very high
yeah, you're
resource
right.
hospital, right? And like there's no reason for that. Like there was no reason to guess that that would have happened with like the care team and how the rest of their pregnancy had gone. And it's just the US in part, obviously medical racism, but also just like how pregnancy is treated in general compared to other places in the world. It's interesting, but we're not going to get into that today.
I mean, yeah, that's a whole PhD in
Right.
itself. But it is a very fascinating topic, and it does get broadly discussed across the year at Yale in particular. And so, just medical equality and focusing on the patient care and patient outcomes, and also just listening to patients is something that I think that should probably be much more hammered into medical school education than it often is, right? Um, so yeah, just listening to the patients. But yeah, I think we can get started now,
So go ahead
and
and share my
yeah, hand it
screen.
over.
Great. I'm selecting the option to have myself overlaid with the background. So you might see me twice, but I just like to feel fancy. So that is why we're doing that. So I'm gonna split today into sort of two sections. The first section, I just want to talk about some sort of general steps in quality control. Some things generally, like some of these principles apply within omics work broadly, but then some are more specific to genomics. And I want to go over these with you because nobody went over this with me when I was starting out. It's something I just kind of had to figure out by myself. And I really did figure this out by trial and error, where like I did something, but I missed a step of my quality control because I just wasn't aware of it. And then I ended up having to redo everything because I had weird samples get through. And that wasn't a good thing. So first, just to like clearly explain what is genomics, because you hear like even I literally right here, I'm just saying omics. Like what is omics? And then what is genomics? So omics is broadly just a measure of some sort of biological thing. So like metabolomics is a measure of metabolites. Proteomics is a measure of proteins. Genomics is a measure of genomes, or in more simpler words, it's just the study of DNA. And so genomics was kind of the first, not the first, this isn't a fair thing to say, but it was one of the first omics to get really big investment and really big government initiatives, just because being able to study DNA really opened the door to a world of research that really wasn't possible before we could study DNA. And the beginning of genomics is before the invention of Sanger sequencing, but I want to talk about or think about genomics since Sanger sequencing. Sanger sequencing is just a specific type of sequencing which allows you to sequence specific regions of the DNA or sequence really whatever based on like marker genes that you supply. It was one of the first, like really kind of widely available forms of sequencing. People still use it today because it's really straightforward, it's really cheap. So for example, if I design a probiotic in the lab and I'm trying to integrate the plasmid into the bacteria to basically confirm that this is a probiotic bacteria that has the DNA that I created in it, I can use Sanger sequencing super cheaply just to see if that thing is there. But now we can get even more complicated. So this is, if I know specifically what I'm looking for, I can use Sanger sequencing, but now I can use what's called NGS or next generation sequencing, and you can get like any DNA sequenced blindly. You don't need to have target genes for about $100 a sample, which is really good. I think when I started my PhD, it was more expensive. I think it's actually even slightly less expensive now. And this is also not the cheapest type of DNA sequencing. So there are some things you can do for about $10 per sample, but you also obviously would get less information with that type of sequencing. I think that within the United States, genomics really became widespread, like not even just in the research world, but also more broadly in the 90s, because in the 90s, the US officially launched and funded the Human Genome Project. And we kind of alluded to this a little bit last time, but the Human Genome Project was just trying to describe what is in the human DNA. So at the time, it was thought that if we understand what's in the DNA, we can understand basically all disease, and we will understand how to solve disease because whatever it is, it's written in the DNA, it's how we respond. And then since then, we found out it's actually more complicated than that. And, we understand, for example, that there's gene expression. So just because we have something in our DNA, it doesn't mean that that DNA is being converted into a protein. You also have things like DNA methylation and epigenetic influences which control which and how your DNA is being expressed. And we're not really going in to talk about that more deeply because I don't work in human genomics. But genomics is really useful for microbiome studies because with microbiome studies, we can't really sequence the bacteria that are there. So microbes that we're testing, for example, from human stool, tend to be anaerobic, which means that they can only survive in the absence of oxygen. Additionally, they're really complex communities. So a microbe might depend on another microbe in order to produce metabolites. And if it's missing this partner microbe, then this other thing that we're really interested in just won't grow. But so now with genomics, it's relatively cheap now compared to what it was even just 5 years ago. And it's pretty accessible. Now they have ways that you can sequence DNA without even really needing a temperature controlled environment. So you can take this on the go with you. Literally, people do this if you need to. It's usually not that extreme though. And it's also very like regularized, I guess you could say. So if I collect a human sequence, or if I collect, let's say, human stool, there's really established protocols for how I'm going to turn that stool into a DNA sequence, so that whether I'm processing this stool, a lab in France is processing this stool, or a lab in the US is processing the stool, we do expect it to be a bit different, but it shouldn't be so dramatically different that we would come to different biological conclusions. So this is one of the benefits of genomics, and the fact that genomics has been around long enough for us to have some sort of standardized methods and some sort of consensus in the field of what is best practice. Yeah. So sequencing methods, this is basically just how am I turning my sample into a FASTQ file is the raw output file, but like how am I turning my DNA sample into a string of nucleotides that I can now read on my computer? So actually I want to go with the second bullet point first. Up until recently, Illumina was really nearly a monopoly over sequencing, at least within the field of microbiome. This was because Illumina had the short read sequencing technology, like they developed the literal machine that you send your sample into for 16S sequencing, for short read sequencing, and also for metagenomic sequencing. And it was when I was like the last year of my PhD, there was another company called PacBio that actually invented a new sequencing method, which to me is crazy. It's like, to have inventions of this type within science and biology, I think really requires a very special mind. So I think it's so cool. Like you have to have a very niche understanding of physics and of chemistry and be able to fit this to new hardware. It's just not how my brain works. So I think it's really cool that people are able to develop this. And so short read sequencing, you basically have some sort of marker gene. So you're telling your sequencer, hey, I'm looking for something that looks like this, go in there and get it. And it's going to give you what's called a short read. So I will get DNA reads that are around 150 base pairs long, like 150 nucleotides long, and that's it. And then I just hope that I get enough 150 base pair long sequences that I can overlap them, and that I'll be able to see the overlap because there are so many sequences. Versus for long read sequencing, I don't actually know off the top of my head how long you can go, but it's, you go from, I'm trying to get as many 150 length sequences as possible to I'm going to just sequence this one specific genome and I can get pretty high confidence that I can sequence that entire bacterial genome completely, like on the strain level, which means that I can tell how different bacteria within the same species are different or the same. We, for our study, are working with 16S short read sequencing. This is the most common type of sequencing method that's used within microbiome studies. And oh yes, Nori sent in the chat, nanopore, I think is in the millions. I think you're right. It's just so different from 16S that it's like a totally different world. And you can ask totally different research questions with the Oxford nanopore compared to the Illumina short read sequencing. So you might choose, and right now, because long read sequencing is newer, it's a little bit more expensive, but it's actually quite comparable now to short read sequencing. So in my last role, both an individual sample to get it either short read or long read sequencing would have been less than $100 per sample. So that's pretty good. And similarly, most 16S studies are shifting away from short read sequencing to 16S long read sequencing, because now it's still a targeted region of the genome, or it's still targeted sequencing. I'm only sequencing the 16S region of the genome, but with the long read method, now I get strain level resolution. Usually with short read sequencing, you don't get to know information on the strain level. You might get to know it on the genus level, but that's really the highest resolution you can get, but you're filtering a lot of reads out. And the reason for this is because we have these 150 base pair windows and we're just overlapping them as much as we can. So it's entirely possible that along the length of our genome, I have 150 base pairs here and 150 base pairs here, and maybe I don't get enough information to be able to map these together. So I can say that this comes from genus A, and this also comes from genus A, but I don't know if these are the same species or different species because I don't have all of this information. So I could have, let's see, actually I should have just put in an image maybe. Bacteria. So 16S, this is a specific region of bacteria, of the bacterial genome. All bacteria have it and only bacteria have it. And it's highly conserved, which means all bacteria have it. It's evolutionarily conserved. And then they have these different regions that are called hypervariable regions. So there's conserved regions, variable regions, and hypervariable regions. The things you might come across when you're reading are them talking about hypervariable regions. So most 16S studies will pick one or two hypervariable regions to sequence, and they basically ignore everything else. The reason that they're only sequencing these hypervariable regions is that it's cheaper. You don't have to sequence everything. I'm excluding the entire genome and I'm making it just down into these two small buckets here. Another reason is that it's usually sufficient for most research questions. So if I can tell what genus is here based on just looking at this small region of the genome, then there's not, and if that is the most important thing, or like I don't need species level information, I don't need to spend the money to sequence the entire thing. So The most common I hear is either V3, V4 or V1, V4. This is a level of detail that you don't need to know. There are some families of bacteria that might be better for like V5, V6 or V4, V6. That doesn't really matter for us. And in fact, that has never been super relevant in my career because human genomics is pretty standardized. So unless you're looking at a very specific disease or you're looking at a specific type of bacteria, you usually just pick the most generic option. And the reason for that is that there are so many things in the microbiome. If we pick a different region, it actually becomes harder to relate our study to previous research because we did things slightly differently. And also these things are the most common, in this case for a good reason, because they're sufficient. They capture enough of the genetic diversity that it gives us enough information while not being overly complicated, versus looking at V3 and V5 might make things a bit more complicated from a bioinformatics perspective. And yeah, so with 16S sequencing, you're only sequencing bacteria. The reason for this is that the 16S region of GNA is unique to bacterial genomes. So that means I don't have any information about viruses. I don't have any information about eukaryotes or yeast or fungi. I don't have any information with host DNA. But from a bioinformatics perspective, this is actually quite nice because now I don't have to filter my reads to remove the human DNA. And even though like it can be really interesting to look at fungi as well, especially like if you're studying the vaginal microbiome, you care about the bacteria and the yeast that live there. You don't just care about bacteria. But most of the time, just looking at the bacteria is sufficient for a microbiome study. So most people are fine with the cheaper 16S version. So cheaper 16S short read genomes.
Emily, just to clarify then, so is the short read decision for the ease of filtering or for the cost, especially since now the cost has been made more, I guess, equal? And my follow-up question was, have you seen in the literature that fungi also are involved in influencing the microbiome? And is this something, therefore, that researchers moving forward should perhaps consider?
Yeah, so for the first question, most of the reason for 16S short read sequencing still being as popular and as widespread as it is is really mostly because that's what people are familiar with. It's cheaper. It's the cheapest actually right now. And there's really like relatively well-established tools. Most people have worked with 16S already before, so they don't have to learn something that's totally new. They can use something that's familiar with them. They sort of know what they looked at before, so they know what looks good versus what would look bad when they do it again. But just as Nori said, so 16S long read sequencing is actually not that much more expensive right now compared to 16S short read sequencing, but it's a different tool. It requires different bioinformatics, even though it's actually really, really easy. So I was able to process 16S long read sequences in my last job, despite having like never done that before. And I got it done within a week because the companies that develop these methods have the bioinformatic tools packaged up so nicely because they really want people to use their tool because they want to be a competitor against Illumina. So I think it's genuinely just people have worked with Illumina for longer. They're more comfortable with it. People are always resistant to change. So they're more skeptical about trying long read sequencing unless they're specifically working with clinical isolates, or if it's really, really important to know very specific information about an individual species or an individual strain, then people are more likely to use long read sequencing. But most of the time, short read sequencing is sufficient. And then the second question was, I don't remember.
The importance of fungi in the microbiome, and have we like seen now that perhaps this is an area that should be layered or integrated?
So fungi are definitely important, but how important they are varies by the body site that you're studying as well as the disease that you're studying. So in general, it's been thought that fungi are only there when things are wrong, that fungi are associated with disease. But again, for example, within the vaginal microbiome, we actually know that fungi are a very important part of the normal, healthy ecosystem. Bacteria and fungi kind of keep each other in check. So the yeast produce a protein called alpha amylase, and the bacteria in the vaginal environment use the alpha amylase to break down other things. So if no fungi are there, then the bacteria can get unbalanced because they rely on the fungi in order to metabolize other things within the vaginal environment. Versus if the so then the bacteria could shrink in population and fungi could become overgrown, and that can lead to a yeast infection. Versus if, for example, somebody has a very high level of stress, the way the body responds to stress will temporarily decrease the population of fungi. And if that population of fungi decreases for a long time, you could get an overgrowth of bacteria that learn, or like they've just evolved to metabolize other things to not rely on those fungi since they're not there. And then you're more likely to get something like bacterial vaginosis. So they are important for health and disease. They're important for maintaining a normal baseline. Exactly how they're important isn't as well understood. Like fungi live in the gastrointestinal tract as well, but human are so variable between each other that it's really difficult to actually know which fungi are important for health. And also the databases aren't as well developed. So I might be able to use a sequencing method to sequence everything that's there. But if, say, 10% of my total reads correspond to fungi, I might, from this 10%, only be able to tell what 30% of those reads actually belong to. Most of the time when you sequence fungi, the majority of your reads are unclassified at the end of the day. This is just because we don't have enough research to understand what's going on.
Fascinating. Thank you so much, Emily.
Sorry, my allergies are picking up, but I was able to work with a fungi researcher. I think the year after we worked together, Nori, in 2023, her name was Laura Tipton, and she's a professor somewhere in the DC area now. But it was really interesting. It was the first time that I learned how important fungi were for maintaining normal health, normal human health. And like how, and you use different sequencing methods when you're specifically interested in fungi, but we're not getting into those.
No, thank you for that insight. My only exposure to fungal has been in a pathogenic sense. And so
Mm-hmm.
um, I was very curious, if we were
Mm-hmm.
now associating them with beneficial outcomes. And that's a perfect example of that.
Yeah. Unfortunately, it's still so, like the databases for fungi are just so poor compared to literally everything else that most of the time we can't do anything with it. So like we can sequence the DNA and we can combine that DNA to say this is definitely a unique organism, but we don't really know what it is. And then if you try to describe what it's doing, so you have these functional pathway analyses, they're all, this is putative protein A, meaning like this is potentially a protein. We don't know what it does, we don't know what it is, but it's potentially there. And they're all just putative proteins. So there's not a lot that you can end up actually doing with that. Okay, so we're using 16S short read sequencing. The full government name for this is 16S rRNA short read sequencing. When I was getting into this, people say all kinds of things. People say 16S RNA, people say rRNA sequencing, people say 16S sequencing, 16S short read sequencing, any combination of these words. I genuinely had no clue if these were all the same thing. They are all the same thing. It took me way too long to figure that out. And the only difference in the microbiome context is what variable regions are being sequenced. But you don't have to memorize that level of detail. Most people, like most clinicians that I work with who are using 16S sequencing, actually just do whatever the biotech company recommends for them. They don't really think about what regions they would sequence. Not because this is good or bad science, but just because they're already very busy. So if somebody comes to them saying, hey, I'll do this for cheap and I think this is the best way to do it, they just say, fine, go do it then. So the sort of general, without getting like too into the details, the sort of general process of 16S is that you have to have some sort of sample that you extract DNA from. So this is a wet lab process and it's quite involved actually. And there's a lot of steps because you have to like dilute your sample, you have to clean it with chemicals, you have to pull out the DNA, you have to check that you actually did in fact pull out DNA and that it's not dirty with other stuff. Then you have to check that the concentration of the DNA is high enough. Then once you've extracted DNA, you amplify the DNA. So the most common technique for this is a PCR or a polymerase chain reaction. This is something that you learn typically if you take like a genomics class or a biology class. So this is something you'll probably come across. But again, I'm trying to not get like too crazy into the details. But basically, a PCR is going to amplify your DNA. And the reason for that is that when you are extracting your DNA, you end up with like this much liquid. Like it's tiny, tiny amounts that you're pipetting into a tube. That's too little to actually give you enough to read. And so you have to amplify the DNA for there to be enough there to actually detect it from the sequencer. Once you amplify the DNA, you send it to your sequencer. Most people work with companies, or most research labs work with companies who have their own machines. The reason for this is it's very expensive to have your own machine, and it's very difficult to maintain. So you have to have somebody who specializes in just that machine. And the technology changes so often that like as soon as you buy it, within 4 or 5 years, it's going to be outdated and you might not get the return on your investment. So these machines cost like $250,000 sometimes, just depending on how new it is that you're buying it and how complicated it is. So most people send their DNA to a company who owns the machine and they pay them to then sequence it. And so Nori has said that molecular genetics labs in undergraduate are going to cover this. Yes. So these are things you might be exposed to in the future. They don't really matter for our internship, but I'm just trying to highlight some key words since you're very likely to be exposed to this more. So once you have your sequencing, this is just the wet lab side of things, but at the end of this, you get what's called a FASTQ file. This is just a raw sequencing file. So you still have to, keep clicking things. You still have to take your FASTQ file and process it from the bioinformatics side. So as well, most people who are going to pay a company to take their DNA and turn that into a FASTQ, they're just going to go ahead and pay the company as well to translate this FASTQ file into an Excel document that they can open. The reason for this is just that bioinformatics can be complicated and intimidating, and it's a totally different field of science. So I specialize in bioinformatics. That means I never do any of these things that are on this screen right now. These are wet lab processes. I don't extract DNA, I don't amplify DNA. My job starts where there's a FASTQ file because it's complicated enough that they need special people just to do that. And so we're not starting from the FASTQ file. We're starting more downstream, but I still think it's important that you're just exposed to what this looks like. In general, you never need to build a data processing pipeline yourself these days because these pipelines already exist. They're really, really well documented. They're open source and free, and they're really good. Like the people who are developing them are the experts in the field, and they're making it freely available. So the only reason you would pay a company is if you really don't know how to process on your own. Like you don't want to learn the computational side and or like you're in a time crunch. So you don't have the time to wait for your students to learn, for example. Most of the time, this is the type of thing that is good to learn This is what can make you marketable. So at a very high level, this is the bioinformatics side of 16S sequencing. So I have a FASTQ file. I know that, for example, I had to use adapters in order to get this thing sequenced. And I was using marker genes to get this thing sequenced because I was specifically targeting the 16S region of the genome. So I want to remove these adapters from my FASTQ file. I also want to remove low quality reads. This is something that happens, I mean, just by chance. You're always going to have low quality reads, but also like if your sample is not very clean, meaning when you extracted the DNA, you accidentally extracted other things as well. So it's a dirty sample. This is really common with saliva samples because saliva is a mucus. It's just so sticky and there's so much stuff in it. It's really, and the DNA biomass is really, really low. So it's really, really difficult to actually get all the other things out and only have DNA left in your sample if you're working with saliva. Then for 16S sequencing, there's two types of methods where you turn those nucleotides into taxonomic information. So the first and the oldest method is called OTUs. And so sometimes, for example, with our own data file, we have a file called zebrafish_otus.tsv, even though we are not working with OTUs. The reason it's called that is just because of old naming conventions. So we still talk about OTUs, not to mean like OTU in the proper way, more just to mean some taxonomic unit specific to 16S. So with the old method of OTUs, you basically had a database of marker genes, and you said, okay, if the marker gene is this, then it's this taxa. If it's this taxa. And then based on how similar your sequences were, so this could be anywhere to 97 to 99% similarity, you would say this is a taxonomic unit. So for example, if something is 97% similar, you might not be able to say that this is the same bacterial species, but you could say it's the same taxonomic unit. So sometimes several species might get grouped together with this method. Sometimes people might group several things at the genus level. But this was really widely critiqued because of this type of threshold. It's really arbitrarily chosen. Like it's not because of the biology, because different species of bacteria are also different. So for some species, there's really high diversity within the species, which means that they would never have 97% sequence similarity, even though we currently classify that as one species. Versus others, they could be very, very closely related. So even at 98% sequence similarity, I would still be able to tell that these are two separate species. So OTUs are critiqued because you have this really arbitrary sequence cutoff that doesn't, it's a cutoff that's chosen for us to make buckets. It's not a cutoff that's chosen because of the biology. Versus you now have what's called amplicon sequencing variants. So without getting too complicated, basically, instead of looking for marker genes and counting the marker genes, I'm just looking within the sequence itself. So when I just look straight at the sequence from the FASTQ file, how many times do I see this sequence? I'm going to count how many times I see this exact sequence. And then you're going to have some sort of factor to account for noise because, for example, you might expect there to be 5% noise, like just normally. So this could account for the fact that like you have higher species diversity, or you might have windows that aren't totally overlapping with those 150 DNA base pairs. Then after you count how many times that exact thing happens, you create buckets and you say, I saw this bucket this many times in the data and this bucket this many times in the data. Now you're going to take those buckets and you map them against the database and you say, okay, how many times did I see this in the data overall? So for most sequencing methods, the second approach is seen as better because it's more true to the underlying data. And so the first one It's called a like mapping based or marker gene based or like database matching type of taxonomic identification, where I can only identify the things I already know and I'm not looking for anything else. The second method, I'm looking at what's there without prior knowledge. So by looking at just what's there and then downstream saying, is this in the database? I can also look at species that I haven't been able to define yet, such as those fungi that we don't have good databases for. Now, fungi we cannot measure with 16S sequencing because the 16S region of the DNA is something that is unique in bacteria. So that wouldn't apply here, but with other types of sequencing like metagenomics, you use the same type of method where you look at what's in the FASTQ file and then see if that maps to a database. This is an area where people are actually investing a lot of money because they think that it's probably the things that we don't already know how to describe that end up being the most important. Then we basically make that into some type of table. Do you guys have questions about this so far? Most of these details you don't need to know, like you don't need to memorize the details. It's mostly for the sake of having you exposed to this. One thing that we're not going to go over with our data, but that I also think is just important for you to be exposed to, is what's called FastQC. FastQC is the most common type of quality control tool that's used in the world of genomics. And I just want to walk through how you would read a FastQC report. I'm closing the chat and I'm going to get the laser pointer. Okay, so FastQC report. FastQC is the name that's given to the report generated for a single sample. And then you also have multi-QC, which is when this report is compiled for many samples, so for a whole study. Generally speaking, because this is the most commonly used report, there's now a lot of good resources online where you can just watch like a 6-minute YouTube video and they'll walk you through how to interpret this. But some of the like biology is also just important for us, so I'm going to go over it quickly. So this is a plot of quality score across the section of a genome. So like I said, you have 150 base pairs. So here we go from 1 to 149, and we are looking at the quality, specifically like the Fred quality, but that doesn't really matter. But the quality of our reads for a specific sample across the entire length of those reads. In general, you see quality decrease over time. The reason for this is just the way the sequencing machines work, for one. Also, just because we can get up to 150 base pairs doesn't always mean that we will get there very efficiently. So we're more likely to see issues with like the sample being dirty, or issues with potentially contamination, or with the adapters doing things to our sample while we're waiting for it to get sequenced. This is more likely to come up when we're looking at the end stage of the genome. And so like I said, our y-axis is a Fred score, but we actually don't have to know that in this case because they label very clearly like what is good, what is okay, and what is bad. So if we had, for example, a lot of our reads ending in this red region, we might be really concerned. I have a real report pulled up. Here. So this is the mean quality score. And you can see like, whoa, these are out of the green zone. They're out of the green zone at the end. Is this okay? This is totally okay. This is normal and expected. Obviously, it'd be awesome if they were all in the green zone until the very end of the sequence length, but that just doesn't usually happen a lot in real life. The important thing is that they're still above this red zone. If they were dipping into the red zone, I might need to go look into these samples and figure out, what do I think happened that impacted the quality of these samples? Like, were they left out on the counter overnight? Were they of a different type of tissue? So maybe the sample was a bit messier. Did we have a different technician processing these samples? Are they from a different clinic? Because then I would just have lower quality and I need to be able to explain that by something. I would still include them in downstream analysis because I know the quality is bad, but I don't know how bad. So I still want to analyze them and see if they look different from the other samples. And then if they look really different, then I might remove them from analysis and redo everything. You also get, just like we saw, this table of general statistics. This is just a really quick look for all of your samples. You can see the percentage of your read that are duplications, the percentage of your reads that correspond to GC regions, or like these G and C nucleotides, and how many sequences in general you got for your entire sample. So first, percentage duplications. So FastQC is something you can use for a lot of different reports. So if you have RNA-seq data, which we are not working with, but it's very common that you're going to have highly expressed genes be more abundant, so your percent duplications is expected to be quite high. Versus for DNA sequence samples, one common method is ATAC sampling or ATAC sampling. This is more common, I think, in gene expression studies, but I'm actually not totally sure. But you expect there to be lower values because you don't have anything that should be so highly overexpressed. So if you have really, really high values, like here in our sample, we see values above 90 and 80%. Higher values tend to indicate over-amplification of the DNA from PCR. And also, it's possible that I'm looking at this FastQC file before I've done any of my quality control steps. So usually you do FastQC before data processing, you do some data processing, and then you do FastQC again, because you want to see, okay, like how many of those adapter sequences did I have that were related to how I was sequencing the technology. Now, I think I removed my adapter sequences. Do I still see a really strong effect of duplications inside of my data? So, but also in general, in microbiome data, you might have a really high percentage of duplications because we do over amplify the DNA by PCR. Like, we know that that's something that we do, but this percentage is quite high from this example. So we also get a percentage of GC content. So GC, again, these are just the two nucleotides. G and C in the genome tend to be more stable. They tend to, like, a specific organism tends to have a really steady level of G and C percentages. So if I'm doing a human genomic study, I can look at the human reference genome, see what percentage of the total reference genome is G and C content, and then create a range of like what I consider permissible, and I would expect this to map that range. If the range is shifted, then this might mean that those adapter sequences are still there that I expected to be out, or in a subsequent plot that we look at. Like if I see really sharp peaks along the regions of the genome in GC content, that's different from what I would expect to see, that could indicate that I have contamination. But this value by itself doesn't mean anything. You need to know what you expect to see from the type of thing, from the type of organism that you're working with. So for example, I was working with human microbiome. So for 16S microbiome studies, you're sequencing a lot of different organisms, basically anything that has 16S, which is any and all bacteria. I actually don't know what the right reference is for GC content when you're looking at a 16S microbiome study. And I worked with clinicians who specialize in microbiome, and I worked with bioinformaticians, and I asked them, and like at that time, we didn't have a good consensus of what the appropriate GC content would be. So for us, because it's not one organism, it's many, it was more about how proportional are these samples to each other. So I see a lot of these are around 50%, which G and C make up half of the total possible nucleotides. So we can say, sure, I guess that's fine. But if I suddenly had a sample that was 90% GC content or 60% GC content, I might go look into that sample more because it's just so different from the other things that are present that I'm not totally sure that that wouldn't be biased in some way. And then if we look at the number of sequences, this is really straightforward. It's just the number of sequences per sample. So here we see all of these samples have somewhere around 200K sequences, but it's not actually the exact same number. And part of this is just, it's really hard to actually control for how the sequencer like really precisely works. We can also see, for example, these are the same sample ID separated by an _1 and an _2. So this would correspond to the forward and the reverse reads. And so also within microbiome studies, you're trying to match these forward and reverse reads to make sure that they agree in terms of like what they actually say is there. So we would hope that between the forward and reverse reads, we're also seeing about the same number of sequences. And we see in this case, this is about true. So again, a level of detail that we're not really going to get into, but drawings, there we go. If I suddenly had a sample here that was only at 100K sequences, this would indicate to me that this might have been a low biomass content sample, meaning there's just a lower concentration of DNA. And maybe I can't actually have very high confidence in the labels that are assigned to this sample because I gave it less material compared to my other samples. I might have to exclude it from further analysis. So for all of these, you really think about them in the context of how different they are from each other. And then it's just if you have specific organisms that you're sequencing or studying, then you adjust what you would expect. Okay, and we looked at this plot. So this is the other GC plot. So this is the percentage of GC content by percentage of what? Why would they do it like this? Sorry. But basically what you're looking for here is really sharp peaks. So a really sharp peak could indicate that you have potential contamination. So if I had something that was like this, so it moves about the same as everything else, but then it had like a sharp peak here and then it went back down. Or let's say it was like really exaggerated. And it was only this one that was peaking up really high where everything else was starting to go down. That might look like a contaminant to me because it's just so different from everything else. So I might want to go isolate that sample and pick it out to see if it looks like everything else. Another thing that could potentially be a contaminant, if I have about the same where everything else is, but then I have like just one really high, but everything else matches about like that. This might also be a contaminant. Just like the trend is the same as everything else, but the actual value just gets so high that I would just want to look into that to make sure that it's the same thing that my other samples are, that I didn't get some sort of weird contaminant. Okay. So you also get, and this is one of the main plots my collaborators ask for when we're talking about our quality control, is they want to look at overrepresented sequences. So overrepresented sequences are just basically, it's really straightforward. It's sequences that are there more than you would expect. So sequences that tend to be there a lot are adapter reads. So again, these are the things I'm using to get my DNA sequenced. So I know that they're going to be overrepresented and I have to filter them out. Some species have there's a list of DNA that's published of things that are just overrepresented for human microbiome. And you need to remove these because it can bias your results, or you have to remove them because they get misaligned. And so we tend to give them the wrong taxonomic ID and we can make the wrong conclusions from our data, blah, blah, blah. So there's always some sort of filtering step for 16S sequencing and other types of sequencing where you're just removing your overrepresented sequences. This is also because of how the PCR works. So anything that's already more abundant will just get more exaggerated. Okay. So now we'll transition to looking at our own data. However, we have been going for about an hour. So I'll give us a 15-minute break since it's also overlapped with your lunchtime. And we can just come back at, I think, will that be 11:15, 12:15? What time is it will it be for you guys?
11.
Okay. And just come back at 11:15 and we'll look at our data. Okay, let me know when you guys are back and I'll share my screen again. Okay, I'll go ahead and share my screen here. Okay. So in our data set, we have a data file called zebrafish OTUs. Here. Let me just pull this up just so we can look at it together. And by itself, this data file isn't very helpful. This is just the raw file, so it's not formatted. But we just see all these ASV labels, and then we see a bunch of numbers that are counts of how many times we count that ASV in that sample. But yeah, this data isn't very useful in the format that it is currently presented in. And it's also, as far as we know, we have to assume it's not quality controlled. We know some quality control has been done by what was published on the GitHub for this paper, but we don't know exactly what was done. And we need to make it into a format that's more useful for us. So we are going to work on some things for it this week. We're going to walk through some adjacent practices during our time together here, but then I'm hoping to give you guys the time through the week to actually try to do it yourself. And then on Friday, we'll sort of go through what the data table looks like and what the Python script looks like to do different filtering steps. So for the quality control, every time you have to do quality control for part of the data processing. So before we even produce the data table, there's a lot of quality control that happens. And we are not doing that type of quality control. This is stuff that happened before we got the zebrafish OTU table. Now that we have zebrafish OTU, there's more quality control that we have to do. And I would say that these types of meta steps really apply to all omics data. So this isn't specific to genomics. It's the little things underneath that are more specific to genomics. So for example, here we need to apply some sort of missingness filter. So that means that I want to filter out here my ASVs, so my taxa, that are present in fewer than 5 samples. Because maybe if it's in fewer than 5 samples, I think it's only there because it's random chance. It's just noise. And also, if it's present in fewer than 5 samples, I don't have enough observations to be able to say anything about it. I can't say if it's different in certain groups. I can't say if it's really there or not, so I'm just going to remove it. Then also for 16S, this is a quality control step that's more specific to 16S, but usually we want to have a count of at least 60. So once again, if we look at our zebrafish, let me draw out those clear out the drawings. So if we look at our zebrafish OTU table, if we just look at these numbers, we see some of these numbers are above 1,000, some are below 100, some are around 100. There's no set golden standard thresholds that anything below 60 is definitely not really there. We don't know. But as a rule of thumb, we can say that we want at least 60 counts of a certain ASV to be present to really have confidence that it was probably really detected. So this would mean that the genome information for that ASV was there at least 60 times within a sample. Then once we do these 2 steps, we want to be able to figure out how many taxa didn't meet these qualification criteria. So we would want to know how many taxa are there before, and then how many taxa are there after each of these steps. So how many taxa were present in fewer than 5 samples and removed, and then how many taxa had counts below 60, so they were removed. After we remove these taxa, not only do we want to be able to describe them, so like in the literal number sense, just counting how many we remove, but we also want to look at those ASV labels. So we want to be able to say, okay, we removed them, but who are they? What do we really think that they are? Does this look like something that's biologically plausible, or does this seem not biologically plausible? So we do this by combining the OTU data with the taxonomy table. And then looking at that, do you notice anything? So not with 16S data, but with, if you're looking at viruses, for example, virus databases change all the time just because we're in an era where we're still learning a lot about viruses. So if I analyze a study of a human sample, sometimes I get a hit for a virus that's a plant virus from my human sample, where I know that biologically that's not there. So if I filter it out, say if it was present in fewer than 5 samples, I might say, okay, that was probably just random noise. Or if it's present in more than 5 samples, but there's not that many counts, then I might say, okay, this might be a closely related species, but it's not really well defined. So we can't say exactly, like it's probably not the plant virus, it's probably something different, but it's just not highly observed enough in our data set to be able to figure out what it is. So this is why it's important to actually investigate what it is you're removing from the data. So you also want to filter out contaminants. In fact, probably you want to filter out contaminants before you apply your missingness factor. That's my bad for putting that in the wrong order. But again, this is something that you do for any omics method. Any omics method, you're always going to filter out possible contaminants. So this, for example, if I'm processing a sample during the winter, I might be wearing a wool sweater. So it could be possible that wool hair from my sweater falls into the sample, and then I could get sheep DNA or something. Like these are real things that happen. You can also, even though you're supposed to be wearing gloves, sometimes people don't. So you can get skin cells that fall into your sample. You can also get chemical contaminants. There's a lot of common contaminants, and usually we don't have to look at this and decide what's a contaminant ourselves. There's published resources. I actually don't have the newest resources because, again, I haven't worked in 16S in a little bit. So I'm waiting for one of my colleagues, my old colleagues, to send me what they use for their contaminants. So I'm just writing this down to also send to you, possible contaminants list, and to send to you by later this week so that you can look at this. Now, because we have 16S sequencing, we're only sequencing bacterial DNA. That means human DNA should not be in our sample. So even if skin cells from human hands fall into our test tubes, we don't expect that human skin cell to end up getting sequenced just by the technology that we're using. So we expect there to be fewer contaminants with 16S, but that doesn't mean that we might not have bacterial contaminants still. And usually it's as simple as you take a list that's been published somewhere online, you see if anything in this list has been found in your data set. If it is, you remove it. Then, just like you do for the first step, you need to look at how many taxa you remove or how many taxa you identify as potential contaminants, and then how many samples that they're actually in. So I will actually add this to the slide. Maybe if it will let me here. How many taxa are possible contaminants, and then how many samples had contaminants. Both of those things are important because if I'm identifying a contaminant in every single sample, that would be, for me, a flag that I need to look deeper into what was going on. It's possible that something happened on the wet lab side, but it's also possible that something happened on the bioinformatics side. Like, am I not filtering my DNA the way that I think I'm doing in my bioinformatic process? Or is my reference genome really old, and so I'm still having these contaminants sort of slip through the net? Just trying to figure out what is going on, because you don't expect there to be contaminants in every single sample. And it's possible it's possible that you genuinely just have contaminated samples, in which case you have to use a really different type of analytical approach, and it's really messy and it's really complicated and it's usually disappointing. So you just want to be as thorough as possible when it comes to the quality control because it limits the type of downstream analysis that you can do. Okay. Then it's always common to transform the data in some way. For 16S, we're converting counts into relative abundance data typically. But for example, in proteomics, you would log transform the data. In metagenomics, it's a relative abundance data, but then you would recalculate a relative abundance after you're applying all these filters. So for all omics, you're doing some sort of data transformation. And so it's actually for metabolomics and for proteomics, you're doing log transformations. Sometimes you also do log transformations in genomics, but it tends to make less sense. And so depending on what our downstream analysis is, we might work with count data, or we might work with the relative abundance data. So we just want to make both of these options available to us because we use them for different purposes. For example, the relative abundance data is going to make a prettier plot, so we use that for plots. However, certain statistics and certain types of alpha diversity metrics are calculated from the counts. So we want to be able to make both of those by the end of the week. And we also have to think about, and this is specific to 16S data, we have to think about how the taxa that are identified at different hierarchical levels would impact our relative abundances. Do you guys have any idea what I might mean by that? Like why that might be important? Yeah, go ahead, Alicia.
Um, Maybe just like thinking about
Hold
if
on, I
we
can't
have
hear you.
different.
But
Oh,
I think it's an
sorry.
issue on my end.
Can you hear me now?
Sorry, sometimes I have to unplug and replug and try again.
Okay, can you hear me now?
Yes.
Okay. Maybe it has something to do with like if we have more of a certain taxonomic level, like percentage wise, and that might be important in our data. And also just considering like if it's not equal, maybe if there's too much of one taxonomic level, that could also be something to look out for in terms of maybe contamination or just like certain levels of things that we want to see and we don't want to see.
Yeah, so you're thinking in the right vein. So from a quality control perspective, there's reasons we want to look at this, but then from a biological perspective, there's also reasons that we want to look into it. And let me see if I can find an example plot for you guys. I should have put one here, but it is okay, because I just used one recently in the class that I'm teaching. So from a quality control perspective, we do want to look at the different taxonomic levels that are there. We want to see if it's relatively balanced, like if is a certain taxonomic hierarchical level dominated by one single ASV, because if so, it can exaggerate how prominent this ASV looks, or like this taxonomic family or category looks in the data. So it's just good to know how many there are in general, but then when you're actually calculating your relative abundance, when you're calculating your relative abundance, you also want to know the taxonomic information, because if I'm calculating an abundance threshold at the layer of the genus, then I have to add up everything that's an ASV that fits this genus. Or if I'm adding it at the family level, so for example, if I'm adding up everything to create a relative abundance at the family level, and I want to know what is the abundance of Bacteroidota, then I have to add up all of these things inside of Bacteroidota to equal the total abundance of Bacteroidota. It wouldn't be correct to only count an ASV labeled here. The reason for this is that it's possible to be more specific than the taxonomic label that we identify. So something could be identified at the genus level, and we're choosing to do an analysis at the family level. So in that case, we just have to make sure that we count these things to be a part of their parent buckets or their parent categories. So if you remember on Friday, when we were looking at ASVs, we found that there was like 200 of them that didn't have a genus level classification. So that means that we would probably look at classifications at a higher level, like the family level, like here, in which case we have to add up all of these genuses or genera inside of the family in order to get the actual family abundance label. And so this plot here is showing you relative abundance data. This is not from 16S, this is from metagenomics. So you can tell because we get more specific information. So here I see actually there's some human DNA that did not get filtered out. So it's the Homo genus, Homo sapiens. But it's at relatively low thresholds, but still this indicates to me that there's more quality control that needs to be done when I'm looking at this plot. I also see like these are non-bacteria or like viruses and that are being identified. So I can know from looking at this that there's other things that are interesting in this plot. But this plot isn't used to show a result. Like Alicia said, sometimes you just want to look from a quality control perspective, what's actually there. So here I'm highlighting two samples. So there's this sample here and this sample here. Do you guys notice anything about them when you look at them compared to the other samples that are presented in this bar plot? Yeah, Adina.
There's just like mainly two colors making them up.
Exactly. There's only two colors. And I know that each of these colors corresponds to some category. So here, they both have this really dark green color, which is Bacteroides. And then one has this teal color, which is probably Klebsiella. Maybe it's E. coli. And then this other one, I can't even tell which green this is, Parabacteroides. I happen to know from human microbiome studies that you would basically never have a human microbiome which only has two taxa identified inside of it. So if I saw this, and in fact I did see this, this was like one of the first plots I made when I was looking at a data set as just part of the quality control. I saw this and I thought, these samples are really concerning to me because they only have these two bars inside of them, or these two taxa inside of them. So when I dug deeper, I found that these two samples were actually low biomass samples, which means that there was a really low amount of DNA that was sent for sequencing. So these are actually low quality samples, and I probably don't want to include them in further analysis because they are biologically different for like erroneous reasons. So I might do this and think that in this case, this is a patient that's had myocardial infarction. And this is a patient that had another type of myocardial infarction. So I might assume that patients with MI have a lower diversity than control patients, when in fact, it would just be these two samples driving that relationship. And it's because of a mistake that happened in the wet lab. So this is why it's really important to look at your plots as part of your quality control steps. I'm not expecting you guys to make this plot by the end of this week because I think that plotting is a little bit complicated within Python. But what we do want to do this week by Friday, and what we will talk about when we'll go over like the results of all of this basically by Friday, are these steps. So try to investigate how many ASVs are missing at different levels. Try to figure out some contaminants. I'll also send something, but like I want you guys to try to think about it for yourselves. But then also, how might we convert our count data into relative abundance data. However, if we saw a plot like what I was just showing you, like this, I might also, because with 16S data, you also get counts. So if I see that there's only 2 taxa in these samples when I look at the relative abundance, I suspect that I actually have a really low number of counts from a 16S perspective. And maybe that can also just emphasize that the reason I see this is because I didn't have that much DNA total present in the sample. So there's just less things detected overall for this sample, like a fewer number of reads for these samples. So, okay. We also, so in addition to these steps, because it's possible that after like our practice, this will be really straightforward, at least the first 2. We can start thinking about what type of descriptive statistics do we think are important. So descriptive statistics are kind of exactly what they sound like. It's just describing the data set. So before we're trying to do any analysis, before we're trying to do any inference, we're just trying to understand what's there in the data. So the most obvious types of descriptive statistics are going to be some things that we already looked at. So how many samples are there per generation per treatment group? We looked at this on Friday using the value counts function, and we talked about how you could make that into a table. You might, now that you have the taxonomic information, be interested in seeing how many taxa after quality control are we detecting per sample on average across the whole study. Then you might want to look at how many taxa are we detecting per sample on average for each generation. What about for each treatment group within each generation? We would hope that these levels are about the same, but if for some reason the treatment was impacting the total diversity of the microbiome, it's totally possible that these numbers could be lower and that would still be like biologically true. There's also statistics that you can use for this type of descriptive work. And we talk about this more next week. This is alpha and beta diversity, but I would say this is more specific to the microbiome world. I rarely see alpha and beta diversity used outside of the microbiome world, versus with all data sets I've worked with, I've always had to answer these types of questions. Just how many is there of what I measured after quality control? How many things did I filter out with each step of quality control? What are the things that I filtered out because of my quality control? These are the questions that I always get asked, both by my bosses and by my collaborators, but also within like conferences and things, because people just want to know that you're really thorough with your work. But also with human disease, which is what I study, sometimes it's the things that are not very common that are driving disease. However, from a statistics perspective, if it's not that common, then I can't actually say anything about it. So people basically just want to know that I'm being thorough with my results. I did look at that. I did confirm the contaminants are actually filtering out contaminants, and I know what the what the types of contaminants are. Okay. So now I'm going to go into, from here, I'm going to go into our VS Code document, or our Python document. And I had to reset this up on this computer. And I'm just trying to confirm that it's working. I have had to reinstall IPython each time I've tried to run this script, which is super cool. But because we're doing quality control, I might want to go into my scripts and make a new file, O1 quality control dot IPYNP. And I want to add a new markdown cell that says quality control. This is our quality control document. We want to investigate, and then I'm just going to copy and paste the things from the slides. And then we'll also make sure that this is here. So then I want to import pandas as PD. That is frustrating. Then I'm going to be working with the same documents that we worked with last time. So I'm actually just going to copy and paste this data over. And here, I'm not going to have this be OTU. I'm going to change this to ASV. The reason for that is that OTU is not a method that we're working with. We are, in fact, working with ASVs. I'm just going to force the restore. The name OTU is arbitrarily assigned by the software that generated these data files because the software is just a little bit older. It has nothing to do with the content of the data file. We've already seen from the file itself that it has ASVs because that is the content of the headers of the cells. And then we also want to read in this taxonomic data because this tells us what those ASVs are. Okay. And then we just have to wait for this to process on my end. But once this processes, we will be able to look at the content of these files. So the first thing that we wanted to do is to apply some sort of missingness filter. So I'm then just going to add another section of this Markdown document called missingness filter, aka controlling for NAs. So if you noticed when I opened our zebrafish OTU file, a lot of those values were 0. That didn't mean it's 0 because it's not there. It just meant 0 because it wasn't detected by the machine. So in this case, we can't treat those zeros like true NAs. 0 values from the ASV table does not really mean that the taxa isn't present. Just that we didn't count it. Because of that, it's really difficult to have enough statistical power to work with the raw data set. So we just have to reduce the number of zeros that we have in the data set. We want to reduce zeros in the data set to try to have enough statistical power to actually use what's remaining. Okay, so I wonder if that finished running, will it let me import pandas? So I don't know that I want to troubleshoot this live because I think this will take a lot of time and this isn't a problem that you guys are having. It's possible actually that I also just need to restart my computer, unfortunately. So basically the problem I'm having is that when I click install IPython, it's not installing it. And then when I go to the command line, so I copy this command. This is my version of the terminal on Windows. And then I paste this command into Windows. Inside of my Python environment, it's not actually forcing the install, or wherever it's forcing the install, it's not being detected within VS Code. So I might have to close it and restart and try again. But I don't want to do that. So for the missingness filter, first, I just want to know how many NAs are actually in the data set, because on the slides, I said that we want the samples or the ASVs to be detected in at least 5 samples, but I don't actually know, like, what if there's no missingness values at all? Like, what if this data set has been pre-filtered? So first, we just describe how many NAs there are in total. So in our case, ASV head, which isn't going to work because I haven't been able to run this code, but we happen to know that this ASV data object has the NAs stored as zero values. So we actually want to count the number of zeros that we have per row or per column. In our zebrafish OTUs table, the TSV file, our rows are individual samples and our columns are individual ASVs or taxonomic units. So this is going to answer 2 different questions. So for rows, if we count the number of zeros per row, this tells us how many taxa we can detect in the study, but that we don't detect in one sample. Versus for columns, this is going to tell us how many samples that taxa is present in. If, for example, this were a study of the human microbiome, the human microbiome is really, really diverse, which means that my microbiome is really distinct from your microbiome, and your microbiomes are also very distinct from each other. We might only have 30% or fewer species shared between us, and we can all be healthy. So, and if I was looking at the human microbiome, I would expect there to actually be, if this is like at the genus level, for example, I would expect that there might be a lot of zeros. Like you might have genuses that I don't have, but at the genus level, this is a higher, so it should be more similar. Zebrafish are slightly less diverse than humans, so there shouldn't be like, these individual samples should have at least like 40 to 50% of the taxa across the whole study detected within them is what I would expect. It's totally possible that it's different and correct, but we just have to check it out. Similarly, when we're looking from the column perspective, I would expect that we should be able to see that these taxa are present in a lot of the samples. Like we said that we only want to look at taxa present in greater than 5 samples, but there should be taxa that are present in at least 10 samples, in at least half the samples, etc. So these are something that we want to look at. Yeah, and Nori said this is really impressive how different our microbiomes are, because she would expect that there might be 60% similarity. It really differs by both the sequencing technique and the taxonomic resolution that you're looking at, but it is like really, really different. This is why it's so difficult to define what a healthy microbiome is, because we're all so different. Okay, so I've kind of already given it away, but if the thing that we want to control for first is how many taxa are present in at least 5 samples, then we know that we care about the column level information. So the way we would think about coding, one way we would think about coding this in Python, because there's a ton of ways to code the same thing, but basically you want to count the number of zeros and non-zero values in each column. That's 2 separate steps. And then you would filter the data by column sums that are greater or equal to 5. So This is how you would execute it from like a meta level, but this isn't telling you exactly what commands you would work with. So I'm going to give you guys 15 minutes to try to work on this. At the same time, I'm going to restart my VS Code so that we can troubleshoot this together. And I'll just send this in the chat just so that you guys have the notes when I stop sharing my screen. And see if you're able to get an idea of how you might count zero and non-zero values for the whole column. And then we'll come back to do this together.
Sounds good.
How are people doing? I haven't finished troubleshooting, unfortunately, my VS Code issues, but you guys can share your screen and we can walk through it. Or do you want more time? Because it hasn't been a full 10 minutes like what we were discussing. I just need some kind of feedback.
Maybe about like 5 more minutes.
Okay, we can do 5 more minutes and then check in again. Okay, so here we go. Python is very mad at me today because even though it worked on Monday last week, it's not working today on my computer. So what we're going to do instead is you guys can share your screen. And I think actually this makes sense anyway, because there's so many different ways that we can code this, and we can try to figure out how to make the solution that you thought of work if it didn't already work. So Adina, do you want to share your screen first? And it's totally fine if it's like not finished or doesn't work yet. We can make it work together. Okay, I'm able to see your VS code.
Okay. So yeah, so for the rows I used this code. So is this like what it's supposed to look like? Or is it, yeah, I don't know. So is this
So
telling me how many rows have the zeros?
So let's look at your code for a second. So you're creating a variable called zeros per row, which looks like it's a series. These are our row index values, and then it's giving you the value 0. So it's counting the things which are equal to 0, because that's what the dot equal function is. Here, it's saying that there's 0, based on everything we see, it's just saying that there's 0 number of values for that row that equal this string that you're giving it. And we can check out our zebrafish dataset, and we think this is probably unlikely because we know that there's a lot of zeros in that data frame. So one thing I would do, so right now you have zeros in quotes, so it's treating this like a float, or excuse me, it's treating this like a string. But then we see the data itself is actually, if we go back to the original data frame and we use something like .describe or dataframe.info, these are actually numerical columns. And so a float will never match a string, even if they're technically the same value. So I would make the 0 inside of your equal function the same 0, just without quotations, and then rerun it and see if it gives the same number.
Okay, I see.
Whoa. So this is saying that like some of these are mostly zeros, which is very interesting, but also totally possible. Again, zebrafish are less diverse than a human microbiome, so we don't really expect them to have everything. So it's possible that all zebrafish just have like 5 microbes, and then there's a couple like, and then another random one that they just acquired in the environment, and that happened with enough fish to add up to 0. So okay, then so this is the sample level information.
Same.
Yeah. So then we're looking at the zeros per column. Okay. And then we see slightly different numbers, but because we only have about 420 rows, so we have some samples are not that common. So yeah, the total number of rows is 421, right? We know this because this is the length of the Boolean that's counting the number of zeros per row. Some of these ASVs have 420. So like if we look at the first ASV, ASV 313, this has 420 zeros present, which tells us that this is only actually detected in one taxa. So one thing we might do is, now that we have this series that has these zeros per column, we might want to filter this list to remove the values that are our total number of rows minus 5, basically using this as a way to say, okay, you have to be present in at least 5 samples in order to be filtered into this list. Do you have an, or did you do more coding steps?
No. Yeah.
Okay, good. Yeah, it's okay. This is a great start. So do you know how we might filter your zeros per column variable?
No, but I'm also like kind of confused. Like, okay, so like I guess just backing up to like what the data table is actually saying.
Mm-hmm.
So like could you explain it a little
Yeah.
bit?
So will you make a new code block? And in your code block, will you just look at the ASV underscore dot head?
Oh, wait, this,
Okay. So here, our rows are these BAP0001. These are sample IDs. So all samples in this study start with the BAP underscore, just indicating that this fish was a part of the BAP study. But this doesn't mean that this was a fish that was exposed to BAP. We have another metadata table, the zebrafish underscore sample.tsv, that tells us the experimental condition information, but we're ignoring that for now. So
so like this is a fish.
that's a single fish, yeah.
Okay.
And then each of our columns corresponds to a taxonomic unit. So this taxonomic unit could be a genus, it could be a phylum, or it could be anything in between. What these ASV number IDs correspond to, like which specific taxa they correspond to, is in the zebrafish underscore taxonomy TSV file. So that's where it has the actual genomic information, but it's just like a mapping file. The OTU, zebrafish underscore OTU table that we're looking at right now is the actual table that's telling us what is the abundance of this taxonomic unit. So this is also what's called tidy data. So tidy data means that each of our samples is one row, and each row should be a unique sample in the data set. And then each of our columns is a feature in that data set. So there should be nothing repeated. So one fish should never take up two rows in the data set because it wouldn't make sense. Similarly, each unique taxonomic unit should only make up one column in the data set. So that's what this data table is. So when we see these ASV 313 or ASV 009, these are just different taxonomic labels. But then to actually see what these labels mean, we have to get the information out of the zebrafish underscore taxonomy file.
Okay.
And then, okay, so now we have these zeros per column and we get the column names with the counts. So if you also look at ASV dot shape or lowercase d with no quotations, this will tell you how many rows and columns you have in your data frame.
Can I do it in the same
Yeah.
Okay.
So this tells us we have 421 rows and we have 536 columns. So we have 421 samples. 421 minus 5 is 416, I think, right? So that means that for our list of zeros per column, if this value is greater than 416, we want to filter out that ASV from the data frame because that means that there are so many zeros that it's adding up to like greater than 5. So we want to take first, I would like to look at the type of zeros per column.
It's like
I'm not sure if it'll work like
Yeah.
that, but you can try it. It might work.
Or did I just literally need to say type zeros per column?
Yeah, but then you wouldn't do this because right now you're trying to execute this as part as asv.dat or underscore dat, but this is a separate thing within Python. So we just want to remove, just fully delete asv.dat. Yeah, Alicia.
Do we have to use the or is it possible to use the is or is null function in this and then sum that up?
Yeah, it is.
Or is that not for zeros?
So the only thing is that here, instead of the NA labels actually being labeled NA, they're just 0. So you just have to specify, if you can, in those functions, you're not looking for NA values, you're not looking for null values, but you're looking for 0 values. And so we see here that the zeros per column is a series. So it has that row index value, and then it also has that count of the number of zeros. So now we want to look at how many values in zeros per column are greater than 416. So if we run this, it should return a Boolean. So it should give us true, false, or something of this sort. Yeah. And so using this Boolean, we can actually now filter the original series because we only want the things that are false. Because the things, we have to think in reverse. We're counting the 0, so we're counting up the number of NAs right now. So if it's true, that means yes, we do want to filter it out because it's mostly NAs. This is detected in fewer than 5 samples. Do you have any idea how we might filter a series?
So we want to filter out the, like we want to get rid of the ones that say true.
Exactly. Yeah, Alicia.
Sorry, I cut out in the beginning when we first got back from like our 15-minute like time. Could you just explain like what we're doing again? Like are we just going through the code that Adina had?
So right now what we're
Like
trying
are
to
we
do,
substituting to get there?
so right
Sorry, go
now
ahead.
we're trying to do is A, describe the total number of NAs in the data set, and B, start applying those quality control thresholds. So the first one was to remove ASVs if they're present in fewer than 5 samples. And so in our data frame, we know that all of our zeros are NAs. So if any individual column, so any ASV, has fewer than 5 values that are greater than 0, it doesn't pass our quality control threshold. Does that make sense? Because that means there's fewer than 5 true observations.
Yeah, I tried to do that and I was trying to use the isNull function or like the isNull function. And when I did that and I filtered out my data frame, I don't have any columns. So I was just wondering like what isn't
Yeah.
working.
We can talk about that after we walk through the example with the Dina's code,
Okay,
because there's like
yeah.
5 or 6 ways that you could do this, just depending on what you already know and what you've been exposed to. And so let's walk through Adina's first. Okay. So Adina has now this Boolean value with true and false that's telling us true, we want to remove this, false, we want to keep this. And this is a series. So do you know how we would filter this series?
Well,
Okay, that's okay.
is there?
Yes, I'm trying
You can use
to.
the log function, right?
You can use a lock function. This would be potentially, well, actually, I don't know if you can do a lock function on a series. Do you want to test it? So Adina, it would be something like zeros per column dot lock all the rows zeros per column is greater than 416. Yeah, but I don't think that this will work on series, but it's worth trying because it's the most. your first 0 per column. If you copy and pasted it from me, it is a B instead of an N. There's a typo. There's multiple typos. Sorry. Okay, so it doesn't like that. Let's look at the error message because we can just confirm. But yeah, too many indexing. Whatever. This is not a very informative error. It's basically grumpy because of how we're feeding it. And it's just because lot can be more complex. It's not quite the same with the series versus with a data frame, but that's fine because there's other ways that we can look at this. What would be the most simple? So what I would actually do is where you have zeros per column greater than 416, I would save this to a new series because this is Boolean information. And I would just save it to something like filter list or like to remove, something for yourself. And then you can do this. Then you would get, how would I do this? Ooh, apparently in the Zoom chat, I can make things look like coding text. It's very advanced. Anyway, it's only useful once I send it to you. You save this as filter list dot values. I'm not totally certain that this will run because I haven't used the dot values function before, but it should work.
Is the E there a typo after the like filter?
Yes.
Okay.
Yes, it's a typo. Sorry, my typos after 3 p.m. like increase incredibly. Okay, so this gives you back a list, but this looks like it didn't work because if we scroll back up to our list above, we see 313 should be filtered out. Or here, wait, we have 313 we want to exclude and 099 that we want to include. Then if we look back at this list, we see ASV 313 and then ASV 330 or 435. So 435 is the next one that has a true value. So in this case, it's returning the values that matches true. So you go from 536 ASVs to 441 ASVs. So now we want to filter this again. So this is the stuff that you would remove. So you would want to save that somewhere so you still look at it. And then we just want to add the squiggly indicates, not this. So then we would just basically want to look at, save the same thing, but by using the squiggly line, we're telling Python everything except what matches this condition. So it should be all of the false values.
Like, this will print the false values.
This will give you another, I think just the column, like just the name of the ASVs that you are including.
So I can save this under like include.
Yeah. And then when you print that, also print out the include list so that we can manually inspect it to see that it does in fact have the values we expect it to have. Okay. So just like we knew from a moment ago, 99 is there. So we expect 102. We see 102 is there. That's good. And then none of those last ones should be there. And they're not there. So that's good. Okay. So just a quick spot check makes it seem like this works. So now we have a list of column names that we want. So now we can use something like asv.lock. Yeah. There's once again several ways that you can do this. And I'm just trying to find a more concise way to do this with all the column names in the list using .lock. The reason I'm struggling is that with the .lock function, you have to specify the name of the column you're working with versus I want to do it for the whole data frame. So I'm trying to think if there is a way to do it by still using .lock. That would be like maybe ASV columns .lock .is include columns. I'm not certain that this will work, but I think this is worth trying. Basically, using .lock to specify the column names are in this list. Only pull out the column names if they're in this list.
You said this is doing what?
This will filter, I'm hoping will filter to give us the data that where the columns are in the include list, include columns list. I had a typo again. ASV column should be ASV underscore dot column. Okay, it's grumpy. Data frame object has no columns plural, sorry. This is why I try not to do live coding demos, but I think they're the most useful for learning. Okay, so now we have a data frame. And again, we're just spot checking. We remember 099 was one we wanted to include, and the next one we wanted to include was 102. And we see that our first two columns are 099 and 102. So now we have data that's filtered to only include samples or to only include ASVs that are present in at least 5 samples. We see this narrows it down from 536 ASVs to 95 ASVs. So that's a pretty dramatic drop. But this is really common with microbiome data. Just a lot of times we count things, but just not often enough. So on your own time, one thing you might want to do is look at the data that was excluded, see how many samples were these things detected in, or like what were these ASVs? So match this back to the taxonomy table to see if it seems like a reasonable bacteria that might be there or might be excluded. But yeah, so this is how we would just filter. This is one way that we would just filter by the data where the ASV is present in at least 5 samples. Do we have questions about this, or can we switch to Alicia's method?
No, we could go to Alicia.
Okay. Yeah. There's so many different ways to do this. So it's totally possible that you thought of a completely different way, Alicia.
So I was just troubleshooting while you guys were talking, and I don't think the ISNA function like was working at all. So I tried to use a function that Adina was using, but I don't understand why, like, what are the parameters that we would put to like filter out the rows and the columns that we don't need? Because I think I'm using the .log function and it is like filtering out some columns and rows, but I'm not sure if it's filtering out the right ones,
Do you want to share your
if
screen
that
so
makes
that we
sense.
can
I
see what
also,
you've done?
yeah, I also didn't understand why we used like 416 at some point. If you could just re-explain that, that would be helpful.
Yeah, I don't remember off the top of my head where it became 416. So we have 421 rows. We can you make your font a little bit larger? We want taxa present in at least 5 samples. So counting the 0 values should be a number smaller than 421 minus 5. So that means if there were more than 416 zeros, then the ASV was detected in fewer than 5 samples. Okay. So I see your first step. Your first step is similar to Adina's, where you're using the dot equal to 0 and then you're counting that up. So now we have a sum of how many 0 values there are for each ASV. And this is column 0 sum, and you have the same thing transposed for row 0 sum, where we have the number of zeros per sample. Okay, now using dot lock, I see. So you're using row 0 sum greater than 60 and column 0 sum greater than or equal to 5. So I'm splitting this out into 2 bits of information. So here, row 0 sum is the sum of the zeros. So you're currently filtering so that you're filtering samples that have more than 70 or more than 60 zeros
So my
observed.
audio disconnected for a second, so I couldn't hear you. Could you just like repeat that again? Sorry.
So your row 0 sum is a count of the total number of zeros you observe for a specific sample. In this case, you're filtering your data to only include rows or samples with more than 60 zeros, versus what I think you want to do is you want to filter the row values by only values where the cell value is greater than 60. So this would actually be a separate step that I would do because you're looking at a slightly different type of math. Does that make sense?
Wait, so is there a way that I could turn like into booleans instead of just pure sums? Like, couldn't I just say here like greater than 60? Like, couldn't I define a parameter here and then get something back?
Mm-hmm. Yeah, you absolutely could. So instead of saying equals to 0, you could basically give a calculation, but the difference is that you want this per cell. You don't want this per row, because then you might exclude an entire sample, even if it's only one ASV within that sample that has an observation lower than 60. So this is why I think it's too complicated to put these together. I think it's better to split them into 2 separate steps, especially because we still want to check the first step and make sure that it's working. So we haven't even looked at the columns calculation that you've done yet.
Wait, sorry, I'm a little confused on like what we're trying to do then. Because I thought we were supposed to be looking at like the rows and the columns and how many zeros there were in each of those and then filtering it out. Like if there were too many zeros, then we would be filtering out the row or the column. Why are we looking at the individual like cells? And like what does that represent?
So I'm going to share my screen and I'm going to, oops, I accidentally removed sharing from Kate. I'm going to replace your current screen share so that you can see my screen share. Okay. So you should see my screen now. So it's loading. Here we go. So within our quality control, there's 2 steps that we want to do. The first step, let's get rid of this and let me move my Zoom window for a second. So the first step that we want to do is filter out if our columns are not present in at least 5 samples. So columns. I'm so good at MS Paint. Columns. Okay. So here, because our NAs are equal to 0, we can just count the number of zeros. So here, we actually have, if we remember, 421 rows, aka 421 samples. If an ASV is present in fewer than 5 samples, then the sum of the zeros for that column should be greater than 416, which is 421 minus 5. So this is the criteria that we're using for the first step, for the filtering. This is using the Boolean and using .log. The second step is something completely different. So, okay. Thank you so much, Zoom.
Sorry, so not to interrupt, but then what is there like a function that we could use or something that says like for each like in each column we can do like I don't know of you know how that we can count the number of like NAs or null values like is there a way that we could count if a value isn't equal to 0 and then if that number in that column is greater than a certain value like greater than 5 then we would keep that we would keep everything that's not greater than 5.
Yeah, you could do this
Sorry,
with
that wasn't a great explanation.
no, I understand what you mean. This is also what I had thought of initially was you can just make or apply an aggregate function. So you apply a function across the whole data set to sum up the number or the number of zeros per column. And basically also all the steps we did, you could make them into one line. So instead of saving the Boolean somewhere separate with the true and false values, you could directly put that inside of a lock statement to filter out the data. The reason that we have this separate is because we want to be able to trace this. And this is our first time coding it, so we just want to make sure that it like we're manually inspecting it, that it's doing what we think it's doing, but also we want to go back and see what we did filter out. Okay. And then, so I also had us count the sum of the zeros for the columns or the ASV, but also the sum of the zeros for the rows. The reason for this is just to see in our zebrafish samples themselves, of all of the taxa that we're detecting, how many taxa are within each sample. So our results had really, really high values. This tells us that we actually are detecting a lot of things, but they're not present in most samples. This doesn't map to 1 or to 2. This is just another general quality control thing, just to understand the spread of our data. So that was just, since we're already counting it for columns, we're also going to count it for the rows or for the samples to understand what the NAs look like there. But 2 is totally different. 2 isn't looking at NAs. Instead of saying, is this value 0 or not, we're actually looking at the true value. We're saying, is this value greater than or equal to 60? Ooh, that's a perfect 6. It totally doesn't look like the 7 or G here. So this is what we want to do for number 2. So in this case, we're looking at each cell of the data. So within each column, within each row, is the value greater than 60? If it's not greater than 60, just make it 0. And then once again, or filter it out. Instead of making it 0, you could make it X, for example. And then you would filter that out because it would be a low quality read. Now, there's no like gold standard threshold on this. So we could use 60, but we might look at the results of that and decide that it's filtering out too much of the data. So we might also test 30. This is perfectly acceptable, especially when you're working with a model organism with a less diverse microbiome. So like this is really common in soil microbiome studies. In fact, this threshold might even be lower in soil microbiome studies. But we as a scientist just have to test both and see if we have enough data to do this type of quality control filtering.
So is there a function or anything related to like the data frame that allows us to go row by row and like check this condition row by row, or sorry, cell by cell? Other and like I guess I was thinking of using like the replace function to replace anything that's less than 60 with like an X or like a 0 or something like that.
Yeah, so there is an apply function. This is, and you compare this with a lambda function or an anonymous function, and it will allow you to go row by row. So axis equals 0 mean it'll go along the rows. Axis equals 1 means it goes along the columns. And then let me stop my sharing. But there's also a lot of ways you could do this. So you could do this with the dot replace function, where if the value is smaller than 60, you change it to something X question mark, whatever, just so that you know that this is a value that you want to filter out. And you just make it traceable this way. So this would be an intermediate step. But after you change it to a question mark, you would then change it to a 0, if that makes sense. It's just for you to see before you change it to a 0, what are the taxa that actually don't meet this threshold. Especially because we need to test a couple thresholds to see, does 30 make sense? Does 60 make sense? Okay. Let's see. We are almost up on the hour, but I don't want people to get stuck. So I will send you untested code because my Python isn't working at the moment in VS Code. So consider this a template of one way that you might look at. Actually, this template code is wrong. I'm not going to.
Is it possible for us to just like try to do it ourselves and then maybe troubleshoot with each other or like put our issues in the chat, in our group chat?
Yes, this
Instead?
was the original intention of having the group chat, but everyone's been so quiet and I've had nobody ask me questions in between our meetings that I'm really afraid that people will run into a problem today and then just hold on to it and not try to do anything before Friday, because I just worry that you guys would get stuck. So I'm going to encourage you to use the chat, and I will send one possible solution on Wednesday. But you guys should also try your own solutions. There's literally 10 ways to do this. Like, for example, you were mentioning using the replace function. I happen to not use the replace function very often just because I didn't know about it for a really long time. So the solutions that I think of don't use that. Both of our solutions are probably correct. It's just a different way to code it. So I I would like for you guys to use the WhatsApp group. And I would also like for you to try to get these 3 quality control steps. So try to apply this missingness filter. I'll send a list of possible contaminants and then try to filter out those contaminants. This is going to involve mapping the ASV labels from our zebrafish_OTU file to the taxonomic labels in the zebrafish_taxonomy file. And then you can start to think about how you might convert this count data into relative abundance data. This last one is a little bit harder, so I don't expect you to have this done by Friday. It's totally fine if you have no idea how to do that. We'll do it together on Friday. But the first 2, I think you should be able to do. For the missingness filter, I'll send a solution Wednesday and then send you the contaminants file Wednesday as well. You're very much welcome to talk with each other before Wednesday, including in the chat, just to see like if you were able to come up with a solution. And then also on Friday, we're going to talk a bit more about like ethics within science. So it's sort of thinking from a bigger perspective and not just with this study, but I'll go over like broadly what is ethics within science, but then also like within my career experience, what are some common ethical problems that I've seen pop up, and how have I seen different people deal with them, and like what do you do in a workplace when they make ethical decisions that don't align with your own professional ethics, because this also might be something you encounter in your work. So that is what we'll go over Friday, and I hope that you will also message me in the WhatsApp because I'm available all week. And then on Friday, actually, I'm not physically here. So I put this in the Google Doc. So we have to either move our Friday meeting to Wednesday, or I can make a video recording and send it. Nori, do you have a preference between these 2 options?
I do not have a personal preference, so I'll kind of leave that up to Adina and Alicia here in terms of also whether they need more time to work on this or whether they'd like to touch up sooner this week on this particular set of code, because yeah, this process is challenging, one, but two, actually being able to kind of go through what options you have available is a really valuable skill for dealing with data in general. So I will leave that up to them too.
What time on Wednesday?
The same time.
Okay, I think that should work for me. Yeah.
I'm fine with Wednesday, but I think I would need more time than just like today and tomorrow to like try to troubleshoot the code and try to figure it out on my own. But I am fine with doing a Wednesday meeting if that's what everyone agrees on.
We can just keep the original plan in terms of the, like when I send the code and the next contaminant sheet, because I do want you guys to have time to actually look at that, because the way you learn coding is unfortunately by struggling through it yourself. So I don't want to take that away because it's taking away your learning opportunity. But I think we should still meet Wednesday because we can just at least talk about the ethics. The author team, I already emailed them, and they're actually typically really good at replying. So I think I'll get a response from them as well by Wednesday. And then we can also go over some of the questions that you guys brought up about the research paper. And then, yeah, next week I'm also not here because I'm leaving on vacation this Friday. And so next week, Monday, instead of recording a video of me talking for like an hour, I'm going to link a couple of short videos talking about like really basic statistics, because I'm assuming that you guys have never really been exposed to statistics before, because at least at my high school, it wasn't required. You could take calculus or statistics. Most people didn't take both. And so we're going to go over just some basic statistics, and then also how would we generate some of the descriptive statistics like alpha and beta diversity for our specific data set. So I'll send all of this out by Friday, just because I don't want to delay next week's work. And I'll also put all the document links in our little living document, the little syllabus rubric that we have. And then next week, you can still text me. I'll just be even more time delayed in terms of when I respond. And yeah, and then I think next Friday from now, Nori had talked about potentially having a single cell RNA workshop, but I'm not sure if that is still the plan or if there's still space for that.
Yeah, I mean, there's still, I have a already curated data set from single cell seq, but since we're coming up quickly on when students will be doing their own presentations, I just wanted to make sure that they also had enough space or like a working day if necessary to kind of go deeper into the data set. But yes, I have a data set that's from our laboratory. It's already super well curated and basically ready to go. And the program that processes it is super visual intuitive and kind of lets you play around with how different genes interact within a very specific context. Again, it's a cancer data set. So I think that if people have the bandwidth for that and don't need support on the coding end for their projects, we can discuss that. Otherwise, we might keep that as a free day just for folks to work on their
Yeah.
projects.